Hello, dear Aquarist. You selected this study because we can learn from nature what can help us to control pathogens, to control fish diseases. And I came across this study on algae, natural algae communities as a potential probiotic for inhibition of aquaculture pathogens. That means also for fish, of course, that we can use something from the nature and specifically here from natural algae that can help in the control of pathogens. And it's a very technical paper, but I will give you the most important details that are important matter for us when we want to take care of ornamental fish or aquaculture fish. Instead of using antibiotics or other chemicals, there is some new pathways we can learn from that we can use bacteria, microbiome from the algae to control a disease like Vibrio. So I will go in detail on it, on the most important matters. So not in detail, I will not give the detail of the study. You will have the link below where you can read the study yourself and all the technical matters in those 19 pages provided in the article. But I outlined in yellow what I would communicate with you. So here, the probiotics offer a promising alternative to antibiotics with the potential to reduce the risk of antibiotic resistance. Because we all know that more and more antibiotic resistance appears due to the large amount of antibiotics used in fish and sometimes badly used in a wrong way. Anyway, discuss this matter that probiotics are typically isolated and used as a pure cultures. However, in natural environments, it is the concerted effort of the complex microbiome that keeps the pathogens at bay. So the nature has a way of controlling pathogens of fish. And here in this study, they developed in vitro an assay to evaluate the anti-pathogen efficacy of mixed algae microbiomes from the live feed microalgae Tetraselmis and Isochrysis. Beautiful names of natural algae. And they measured the inhibition of fluorescent protein from the Vibrio, so they can measure it on a laboratory way to see that it is as a key that Vibrio is an important fish pathogen, particularly as we know in marine fish, that the microbial communities were measured and quantified as a reduction in fluorescence. That means they have an impact on the Vibrio, on the bacterial infection. This study demonstrates that the microbial communities derived from natural algae microbiomes can have anti-pathogen effect, and that the bacterial co-cultures may offer synergistic advantages over monocultures as probiotics, highlighting their promise for aquaculture health strategies. I will go more in detail on that during this study, but they use different methods, and I explain what they did to prove the efficacy of those microbiomes from these algae. You can go in detail with, I just scroll through it, what they did, how they did it, how they identified it. And here is this scheme, what they did. So they had here in A the algae cultures. The xenic culture are those with the microbiome, and the axenic cultures are lacking the microbiome. They also multiplied here above the microbiome to put it on plates where you can also put the Vibrio bacteria on them to see how does microbiome act or inhibit or control the Vibrio bacteria. And that the examples of different cultures with the algae, without the algae, just the simple microbiomes, or those without microbiomes. And then they put it also on agar plates and they see the inhibition. And that's the thing that happens in a laboratory when you do sensitivity testing to check if an antibiotic works properly on a bacterial pathogen. They can see the inhibition, and that the inhibition says, well, it means that this antibiotic, or here today, those bacteria from the microbiome can have an effect on the bacteria, controlling it. So they will go in detail to give the results later on, but I go further through. They did sequencing, well, very technical, but I want you to give you the most important details here, that you can see here the growth of the Vibrio that was diminished. You can see less growth over time, and that means there is some impact. Well, not go in detail, but this study tells you that. And here they show you the different algae species, the isochrysis, in comparison to the tetraselmis. And we could see that the isochrysis has a better impact on, how can I say, on the inhibition of the Vibrio bacteria. And this is the impact of the different type of microbiome. And this is not so great with the tetraselmis microbiome. So the isochris microbiome has a better impact on the Vibrio. This explains it, that the effect of the different algae. And then they went to study the different bacteria from the algae. And here is the normal, on the left here, the normal group of microbiome from that, well, isochrysis. And here it's enriched. So they have used a method to enrich that microbiome, to cultivate it, so they have more potential to use it as a natural antibiotic. Just to try out in vitro, what can we use from our algae? And then they did testing on it, on the Vibrio, on the inhibition with pure cultures and co-cultures of bacterial isolates. And they used pure cultures of all 64 bacterial isolates, and they were tested if they inhibit the Vibrio. And it was clearly that 17 isolates had a pronounced inhibitory activity, and most of them were identified as Phaeobacter. That were the most natural bacteria from the microbiome that was active against the Vibrio. And here you can see the results, the positive impact in inhibition, mainly due to Phaeobacter microbiome bacteria. And here you see it on the sensitivity plates, on the testing, and they put out the Vibrio on the plates, growing on an agar culture. And you can see here the sensitivity discs. You could see the H2, everything where H2 was added onto it, particularly, there was a diminishing, how can I say, less growth of the bacteria, Vibrio. Also D3 here had an impact, but particularly we concentrated on the H2 in general, that had a major impact on, well, can I say, on the Vibrio bacteria. So they made a conclusion that the study was clearly that the microorganisms associated with these algae can inhibit the growth of a pathogen, emphasizing the crucial role of the microbial communities associated with live feed in larviculture for disease control. because those algae are usually fed to live food, and the live food is given to the larvae and can help in this case to apply a natural antibacterial treatment. And this highlights the potential of the harnessing of natural microbial consortia to enhance disease resistance in aquaculture settings. For everybody dealing with breeding fish, it's an important way to look at this potential of this study. And that's why I want to emphasize this. In conclusion, by introducing a good method for screening mixed microbiomes, this study offers a promising approach for identifying candidate microbiomes for sustainable disease management. And these findings highlight the potential of harnessing natural microbial consortia to enhance disease resistance in aquaculture and support the importance of microbial co-cultures as potential disease control strategies. Well, this teaches us a lesson. I wonder why our fish love to eat in nature natural algae, usually incorporated in live food like Daphnia or Artemia, particularly in saltwater, the little invertebrates that the fish eat, and they consume algae, microalgae, and also the potential bacteria that had a real good effect, in this case, in the study on Vibrio. So nature takes care about his fish. The fish know how important food is, and we should also learn from these kind of practices. And I think keep an eye on these developments, how important the algae can be as a nutritional food source, but also as an antimicrobial, anti-pathogenic potential. Subscribe and learn more from my studies if you want to become an advanced aquarist, particularly the breeders. Thank you for watching.